Now for a quick discussion about the versatility and regulation of photosynthesis. There's an evolutionary quirk with photosynthesis. It turns out that the enzyme rubisco, the one that we talked about that is very important for photosynthesis, it evolved in conditions of low oxygen. Okay, so it evolved in anaerobic conditions. Therefore, as a result, its active site, so where it normally binds to a substrate, has a very high affinity for oxygen gas. So although rubisco shouldn't bind oxygen during the process of photosynthesis, it can. And as a result, we have something that happens called photorespiration. Okay? During photorespiration, the metabolic pathway that occurs when rubisco incorporates oxygen. So it's when rubisco binds to oxygen instead of carbon dioxide into RUBP. It is a metabolic dead end. It uses up ATP, but it doesn't produce any sugar. So therefore, it's basically just wasting ATP. So it is best if avoided.
All BT1s, please report to the media center. All BT1s, please report to the media center.
Sorry for that little announcement. But lots of times that photorespiration is not a problem. As long as the plant can keep its stomates open and keep exchanging gases with the environment, photorespiration is kept to a minimum. Okay, so remember, our stomate is the very little hole that's in the bottom or the top of a leaf, and it lets gases pass in and out. Okay, and remember, our guard cells control whether or not that stomate is open. So if our leaves live in an environment where it's able to keep its stomates open, then it has no problem with photorespiration. We have these plants called C3 plants that have no adaptations for minimizing photorespiration. They live in environments where they can keep their stomates open. Okay, both stages of photosynthesis for these plants occur in the same cell simultaneously. Okay, oxygen and carbon dioxide are exchanged with the environment through the stomates pretty much at all times. Okay, they can pass through the stomates. And sugars are going to be transported to the vascular tissue throughout the plant. Okay, so it has our C4 or C3 plants. C3 plants have no special adaptations to prevent photorespiration. But sometimes there are environments where it's dangerous or it's harmful to keep stomates open. It will lead to drying out or lead to desiccation. Desiccation is just the fancy word for drying out. Okay, so living in a dry environment leads to desiccation unless you actually close your stomates. When you close your stomates, that leads to an increasing oxygen gas concentration as produced by photosynthesis and a decreasing carbon dioxide gas concentration. Remember, when we put brackets around something, that signifies concentration. And when we have increasing oxygen compared to decreasing CO2, that leads to increased photorespiration, which is not good. So plants are going to have some adaptations to avoid this. There's two major adaptations. Our first type of adaptation is found in C4 plants, C4 leaves. They have a spatial separation, which helps to prevent photorespiration. Then in our C4 plants, carbon fixation occurs in the mesophyll cells. Okay, so you're fixing carbon in the mesophyll cells, and carbon dioxide is incorporated into a 4-carbon organic acid called malate by the enzyme PEP carboxylase. And PEP carboxylase binds to carbon dioxide, and PEP carboxylase has a very low affinity for oxygen. Its active site doesn't bind oxygen under normal conditions. Okay, so we're making this 4-carbon acid, and then we transport it to bundle sheath cells in the leaf. Okay, and the bundle sheath cells is where the carbon dioxide will then be cleaved or broken away from the 4-carbon sugar. Okay, and since the bundle sheath cells are surrounded by mesophyll, they're not exposed to as much oxygen. So their oxygen gas concentration is going to be lower. Okay, and as a result, so their oxygen is lower, and even the light reactions can occur in the mesophyll cells. So we separate where these things are happening. Light reactions are going to happen in the mesophyll, and then our light-independent reactions can happen in the bundle sheath cells. Our other type of adaptation is seen in CAM plants, CAM plants, and they separate when they do the light and light-independent reactions in a temporal separation, not spatial separation. So carbon fixation occurs during the evening, okay, when open stomates will not lead to desiccation. So they open up their stomates leave them open so that we can exchange carbon dioxide for oxygen and oxygen for carbon dioxide, and we can fix carbon then. But then carbon dioxide is stored as an organic acid. So we take in that carbon dioxide during the evening in our open stomates, and then we store them as an organic acid. Then we close our stomates during the day so they don't dry out. And the organic acid that was stored is used to supply the Calvin cycle with carbon dioxide. So now let's discuss the importance of photosynthesis. If it's not obvious to you already, consider that everybody you know and every ancestor you've ever had, they all were able to exist because photoautotrophs, or plants, or other things that use photosynthesis to produce sugar, all those plants make more food than they need so that we can eat them, or other animals can eat those plants, and then we can eat those animals. All right. Also, modern industry is more and more interested in using plants to do all sorts of different things. Okay, we can manipulate plants to produce other things we'd like to use. We can use biotechnology to do that, or we can use plants to make biofuels to help power our cars instead of using fossil fuels and coal and things like that dug up from the ground.